Ladies and gentlemen, my name is Eustace Ehiwe Matuda, and today I'm going to be talking about microorganisms. Microorganisms are tiny organisms that cannot be seen with the naked eyes, only with the aid of a microscope. The branch in biology that involves the study of organisms that are microscopic in nature is known as microbiology. And microorganisms can be found everywhere, including on air, water, soil, on surfaces, on our bodies, on our clothes, and even inside our bodies. Microorganisms are grouped into 5, which are virus, bacteria, protozoa, fungi, and algae. Now, talking about viruses are very small that they cannot be seen with the aid of a normal microscope, but only with the aid of an electron microscope. For virus, they are divided into 2, which are the RNA virus and DNA virus, and they can either be enveloped or not. When you mean they are not enveloped, it means they are naked and can only reproduce inside a living cell. Now, going on to bacteria. Bacteria lack nucleus and they contain cell wall, cell membrane, slimy capsule, and a dense cytoplasmic granule. For bacteria, they can be grouped using these 2 things. Firstly, with their use of oxygen, and secondly, with their shapes. For the use of oxygen, we have aerobic bacteria. From the name, it means that they can only survive in conditions that have oxygen. And for anaerobic bacteria, they can only survive where there is no oxygen. And lastly, for facultative bacteria, they can survive in places with both oxygen and no oxygen. So they can survive in aerobic and anaerobic conditions. Now, going on to bacteria grouped using their shapes, we have cocci. Cocci are circular in shape, and they can either be clustered together, which is staphylococci, for example, boibacteria, or they can be in chains, which is called streptococci, for example, sore throat bacteria. Now, for bacilli are rod-like bacteria, and for bacilli, some of them contain flagella or flagellum for movement. An example for bacilli is the bacteria that causes typhoid fever. Now, going on to vibrio. Vibrio are like bent in shape, and an example is cholera bacteria. Lastly, we have spirale, and they are twisted in shape or spiral in shape. An example of spirale is syphilis bacteria. Now going on to protozoa. Protozoa, from their name, they can also be called protista or protists, and they are single-celled organisms. You can also call them unicellular organisms. Some are parasitic and some are not. For the parasitic ones, we have trypanosome and plasmodium. For example, for trypanosome, it causes sleeping sickness or trypanosomiasis, and for plasmodium, it causes malaria fever. And for the harmless ones, we have unicellular organisms like amoeba and paramecium. Now going on to fungi. For fungi, they are not green plants. For fungi, they are either parasitic or saprophytic. For the parasitic ones, they cause different diseases, and for the saprophytic, they are beneficial to our environment. We also have algae. Algae are green plants, and for algae, most of them are found in aquatic habitat. For example, Chlamydomonas, nostoc, diatoms, oscillatoria, Spirogyra, and Volvox, to mention but a few. Now, going on to culturing. Culturing is when you prepare a sterile medium and grow microorganisms. And for culturing, it involves preparing a sterile medium, inoculating, incubating, and examining the features of the organism. For the features, we have like how they grow, their pattern of growth, their color, and different things to examine. Now, here are some apparatus that can be used for culturing. We need a petri dish, microscope slide, inoculating loop, inoculative chamber if it's available. We also need test tube or test tube holders, to mention but a few. Now that you have seen this video, please like, comment, and share, and don't forget to subscribe to our YouTube channel so that you can be involved whenever videos like this come up. Thank you.